Thank you to the organisers of the conference for accepting this abstract as a presentation. What I'm going to present to you today is how we developed a hybrid clinical research model of what we're calling patient choices in genomic medicine for implementation in the NHS Genomic Medicine Service. This was based on the work we did in the 100,000 Genomes Project. As many in the audience will know, in the UK 100,000 Genomes Project, participants consented to whole genome sequencing, return of main findings, findings related to the reason of recruitment, and opted in or out to the return of a limited list of additional findings. Participants were recruited from the NHS. They were patients who were attending clinics in search of a diagnosis for their rare disease or patients with a specific cancer diagnosis. The consent materials were created and revised through an iterative process. It was done in collaboration with participants and recruiters. Genomics England has an independent ethics committee and a participant panel, and of course it went through the appropriate ethical and governance approvals. And all the materials are available on the Genomics England website. In learning from the 100,000 Genome Project, we learnt about the process of implementation, but also other people have done research and looked at the experience of healthcare professionals and participants. And I present two extracts from two different papers. One focuses on the need for workforce preparation, is that health professionals needed to be prepared to answer patients' questions in order to manage the consent conversation appropriately. The second extract talks about the reasons why participants took part in the 100,000 Genomes Project. It was not so much based on the facts of the project, but there was an element of interpersonal and institutional trust in the NHS, and they considered they were making an investment in improving care for the future. These are important factors to consider when we are going forward, and it is really also important not to damage that trust. So what we also learned from the 100,000 Genomes Project was, of course, it is difficult to maintain consent status, recording of consent status at scale. We recruited over 80,000 participants, and all of those participants should have an entry in our data systems that records their consent status that is accurate at this moment in time. Consent choices may change over time. Of course, they may withdraw. And for the 100,000 Genomes Project, that is partial or full withdrawal. For additional findings, they could have originally said yes and then changed to no, and conversely, no and then to yes. And of course, participants could either gain capacity or lose capacity, as it is a lifelong involvement in the project. And a number of the participants were children, and of course, many of them will gain capacity as they reach maturity. This is not a simple matter to design a system that maintains this at scale. And our learnings from the 100,000 Genomes Project have fed into how we have implemented the patient choice work in the Genomic Medicine Service. So in the Genomic Medicine Service, which is a total system transformation of the way genetic health services are delivered in the NHS in England, predominantly focus on the delivering of tests. The aim of the patient choice aspect of this is to give a clear, informed and separate choice about the genetic test, and initially this will be only those involving whole genome sequencing and participation in research. But the offer to consider participating in research is part of the clinical pathway, and all patients where whole genome sequencing is used as the test technology are expected to be given the opportunity to participate in research. The research relates to access to their data and potentially samples in what we have called the National Genomic Research Library. And this is access to de-identified data by researchers from both for-profit and not-for-profit organisations, recall for other research projects, lifelong collection of other routinely collected health data, which is imported into the data environment, and it can also include samples. In terms of the clinical choice, the tests on offer as part of the Genomic Medicine Service are NHS commissioned diagnostic tests for patients presenting with unexplained symptoms, and the consent conversation with these patients is unchanged from current consent standards. Whole genome sequencing as a technology that is used for some of the clinical indications is commissioned centrally depending on the clinical indications for doing the test. What we are explicit about is that the data will be stored nationally, and that for the limited number of cancers where whole genome sequencing is being used on a somatic basis, that there will be discussion of pertinent germline results. We called the consent conversation patient choice, as really it is a choice about the genetic test combined with a choice about saying yes to the research offer. So in terms of the clinical aspects of the genomic test, the recording of consent is based on a record of discussion, and this is in line with the consent and confidentiality work of the Joint Committee on Genomic Medicine and on Genomics and Medicine. We originally were intending that the recording of the conversation would be done electronically and would be patient-facing. Not unexpectedly, the ability to integrate the patient choice module with the rest of the IT systems across the NHS in England has proved complicated, and currently we're using an initial paper-based systems. So on this slide, there's the first page of the paper-based form, and I've pulled out 3 statements which I think are considered to be a bit novel. And this is again based on work in the consent and confidentiality document. So it is explicit that the results will be held in a central database, and that the results of one patient will be used to help interpret the results of other patients, and their results will be used to help interpret the patient's results. In addition, if results have familial implications, then those results can be used in the testing of those family members. This will be done in a process that won't identify the original patient, or if there are separate issues, then this could be done in discussion with them. Also, in this first clinical facing record of discussion, it is clear that the opportunity to say yes to research will be presented to the patient. The second page is the participation in the National Genomic Research Library. This is what we have called the data resource that is being created. The statements relate to security, recontact, data and sample usage, data storage, and withdrawal. It is backed up by other resources, both online and in print. And as before with the 100,000 Genomes Project, this is an iterative process of creation, almost co-creation with participants and NHS professionals. We are looking at the version that was developed for, as we say, minimal viable product 0.0. The third page is the confirmation of the genomic test and research choices, and the patient signs to indicate they've had the discussion, and they circle whether they want to contribute to the research or not. As you can see, it is a fairly simple form, and there are no tick boxes. So other considerations for implementation. The 100,000 Genomes Project was a complex project within the healthcare system, but it was regulated as a research project, and whole genome sequencing was done under research governance. In the Genomic Medicine Service, we have a combination of a clinical service under clinical governance and a research offer under research governance. It has been a whole system transformation involving reorganisation of all the genetic laboratory services in England and the creation of the National Test Directory. Genomic tests will be ordered through one test ordering system, and this gives the opportunity for the patient choice module to be attached to that test ordering system. So alongside the information about the test order will be recorded the patient choice for research, and this is auditable. This has also needed a programme of workforce transformation, which has been delivered by Health Education England Genomics Education Programme. Their website is here, and they have developed an enormous menu of educational resources related to offering genomic medicine within routine care, as the intention of the Genomic Medicine Service is to gradually mainstream genetic testing as appropriate and as clinically indicated. We had intended to have data to present to you when we submitted this abstract. Of course, we're now in a new world, and COVID-19 delayed the start of the Genomic Medicine Service, so as yet we don't have any of that data. However, the development of this simplified approach to obtaining consent for both clinical testing and research testing has been essential to collaborations in relation to COVID-19 research. All the materials and the approach have, of course, been reviewed by research ethics committees and are in line with regulatory, ethical and governance principles. This has enabled us to enter into partnerships with other studies, providing the data environment and the host genome sequencing for those studies, and collaborations are ongoing. We would not have been possible to do this quickly in the way that we have if we hadn't had this consent model already developed and starting to be implemented. So of course, I'd like to thank everybody who took part in the 100,000 Genomes Project, both participants and NHS workforce. My colleagues at Genomics England, NHS England and Health Education England, and my colleagues at the Wellcome Genome Campus who support part of my salary. So thank you.